Hereby I open this academic ceremony in which Stefano Navarro will defend the academic thesis Novel Platelet Glycoprotein 6 and CLEC-2 Targeting Strategies, Studies in Humanized Mouse Models. May I invite you to present a summary of your studies and the conclusion of your thesis.
Thank you. Dear Prorector, dear members of the Corona, dear family and friends, dear audience, thank you very much for being here today. In the next 15 minutes, I'm going to give you a brief overview over the main findings of my thesis, which is called Novel Platelet Glycoprotein 6 and CLEC-2 Targeting Strategies, Studies in Humanized Mouse Models. First of all, I would like to give you some introduction. If we take a look at the official website of the World Health Organization, we can immediately see that the two leading causes of death globally are heart attack and stroke. And this happens in part also because the currently used drugs work incompletely and often have side effects. GP6 and CLEC-2 are platelet receptors. Platelets are blood cells known for their role in the process of thrombosis and hemostasis. Thrombosis is the process that leads to the formation of a blood clot in a vessel. The process of hemostasis is often represented as a scale, as an imbalance within this process may lead to different kinds of diseases, such as if we have a reduced hemostasis, we may have an increased risk of bleeding. Whereas if we have an increased hemostasis, we may incur an increased risk of thrombotic events, such as stroke, heart attack, or lung embolism. If we take a closer look at a platelet, we can immediately see that it expresses a pool of receptors on its surface. Receptors are specialized proteins that are expressed on the cell membrane, and they help the cell interact with its environment and to respond to different signals. In my thesis, I mainly focus on the study of these two receptors here, which are called glycoprotein 6 or GP6 and CLEC-2. They are receptors for platelet activation, and they do so by binding to different molecules. However, they share a common signaling pathway. In my thesis, I mainly focus on the study of the receptor glycoprotein 6. This receptor is the main receptor for collagen on the surface of platelets. Collagen is a molecule that gets exposed upon vascular damage, but also upon atherosclerotic plaque rupture. Interestingly, when GP6 enters in contact with collagen, this leads to the activation of platelets and to the stimulation of thrombus formation. If we take a closer look at this image, we can immediately see that the process of activation behind these two receptors is rather complex. However, this leads to a few major events in the cell body, which are the change of platelet shape, the secretion of their granules, and the activation of a specific kind of receptors called integrin. But let's take a closer look at this process. Upon vascular damage, platelets can recognize the injured vessel thanks to a pool of receptors. They start rolling on this diseased vessel and get activated. The activation can happen through different mechanisms, one of which is the recognition of the molecule collagen via the receptor GP6. This leads to full activation of platelets and the activation of the integrin receptors, which are necessary for the stable adhesion of cells to their environment, and the release of platelet granules, which contain proactivatory molecules that allow to recruit the other platelets to the injured vessel and to further activate platelets in order to stimulate the formation of a thrombus or an aggregate. Together with the process of platelet adhesion and activation, we have the coagulation cascade, which is also a rather complex mechanism that has as final event that of generating fibrin clots. And it does so by forming a molecule called fibrin, which is a very sticky molecule that allows the stabilization of the growing thrombus. But the coagulation cascade does more than that. It also generates a molecule called thrombin, which not only helps with the generation of fibrin, but it also activates platelets itself. Now, in my thesis, I mainly focus on the study of the receptor GP6, as this has been for long proposed as a promising antithrombotic target. Previous studies already showed that anti-GP6 molecules have an antithrombotic potential, but also, also that a lack of GP6 or pharmacological inhibition of it does not cause major bleeding complication. Interestingly, first inhibitors of the receptor GP6 are finally entering the clinic. In my thesis, I used a series of tools that interact with platelet activation and coagulation. However, for the purpose of this talk, I will mainly focus on the two major tools that I have used. These two tools are two antibodies. Antibodies are blood proteins that our body produces in order to fight infections. And they can recognize specific sites on specific proteins. The first antibody that I used is called JAK1, and this binds to mouse GP6 and blocks receptor function. The second one that I used is called instead EMF1, which binds instead to human GP6 and also blocks receptor function. Now, in my thesis, I had various aims. However, there were three major questions that I wanted to answer with my work. The first one is, what is the effect of the antibody JAK1 on human GP6 in vitro? The second is, how long is GP6 signaling lasting during the process of thrombus formation? The third one is, can we target human GP6 using the antibodies EMF1 and JAK1 in vivo? In order to answer some of these questions, I had to use another tool, or better, a humanized mouse model. Humanized means that these mice were genetically modified in order to allow selective expression of human GP6 instead of mouse GP6 on the surface of circulating platelets. This modification also allows us to study potential medications directly in vivo. Let's go now to the first experimental section of this talk. We wondered, what is the effect of the antibody JAK1 on human GP6 in vitro? In order to do this, we took blood from either control mice or humanized mice and treated it with JAK1. Treated blood was then perfused over collagen with a microfluidic system in order to assess the rate of aggregate formation. If we do this with blood from control mice, we can immediately see that the JAK1 binds to mouse GP6, inhibits its function, and abolishes aggregates formation over collagen. However, if we repeat this experiment on blood from humanized mice, JAK1 binds to human GP6, but instead of inhibiting its function, it enhances it. And this leads also to an increased aggregate formation over collagen. Among the various reasons why we believe this to be interesting, this highlights the suitability of mice expressing human targets to test novel medications. The second question we wanted to answer was, for how long is the signaling of GP6 lasting during the process of thrombus formation? In order to do this, we had to modify the currently used Maastricht flow chamber, which is a microfluidic system, by a 3-tube inlet system that would allow us to switch from untreated to treated blood without having to stop the blood flow. If we take a closer look at this system, we can see immediately that the blood is immediately allowed to mix with the recalcification buffer that allows proper coagulation. These systems makes it easier for us to test time-dependent pharmacological intervention. Therefore, we use it to treat human blood using different treatments at 2 different time points. Either at time 0, which means before the formation of thrombi has started, or at time 2, which means 2 minutes after the formation of thrombi has begun. We also measured a series of measurements for platelet activation and coagulation during the process of thrombus formation. In my thesis, I used and tested a series of treatments. However, for the purpose of this talk, I will only show you the results with the antibody EMF1. We can see immediately from these images that if we treat human blood using the antibody EMF1, we have a very strong reduction of thrombus formation and stability under flow, but also an abolishment of the activation of platelets and a very strong reduction of fibrin deposition. However, if we treat the human blood with EMF1 2 minutes after the formation of thrombi has started, we completely lose the effect of this antibody, which tells us that GP6 inhibitor seems to be ineffective at later phases of thrombus formation. The last question we wanted to answer was whether we could use these antibodies, EMF1 and JAK1, to target GP6 in vivo. We do this because previous evidence has shown us that when we inject JAK1 in control mice, we have a complete removal of the receptor mouse GP6 from the surface of circulating platelets, which leads to protection of the mice from thrombotic events without causing major bleeding events. Therefore, we treated the humanized mice using the antibody JAK1. But we found out that injected JAK1 can only partially remove human GP6 from the surface of circulating platelets. Nonetheless, this was still sufficient to strongly reduce aggregate formation on collagen underflow. Based on this unexpected finding, we sought to assess whether we would be able to remove completely human GP6 from the surface of circulating platelets. Therefore, this time we repeated the same experiment, but we injected the humanized mice using the antibody EMF1. And we found out that the injection of this antibody completely removes human GP6 from the surface of circulating platelets, and that this leads to a complete abolishment of aggregate formation under flow. So, what did we learn from these experiments? First of all, we found out that the antibody JAK1, which is known to inhibit mouse GP6 function, it instead enhances the function of human GP6. That GP6 seems to be pivotal mainly in the early phases of thrombus formation, and that in mice, even partial removal of GP6 can act as an antithrombotic, which is a promising result for the study of treatments for thrombotic diseases. With this, I come to the end of my talk. I would like to thank all the members of my groups, both here in Maastricht as well as in Würzburg, all the members, all my supervisors, Professor Bernard Nieswand, Professor Johan Hemskerk, Dr. Marijke Kuipers, Dr. Heike Hermans, all the members of the Corona, and all of you for your attention. And I hand off the word back to the prorector.
Thank you very much for this presentation. It's now time to continue with the opposition. And the opposition will be opened by Professor Koene. He's Professor of Vascular Inflammation and Thrombosis at Maastricht University.
Thank you, Prorector. Dear candidate, I would really like to take the occasion first to give you my compliments on this great thesis. I've read it with a lot of interest, and I really especially enjoyed the introduction, which was very comprehensive and well written. I also like the translational character of your work and of your thesis. And this, of course, includes my congratulations to the supervisor team online and on-site. So I've read your introduction, and I have the impression that GP6 is a real, to say it in German, Tausendsasser, which is a person that is actually can do everything. So it's almost involved in every aspect of thrombosis. It's in a lot of platelet functions in platelet leukocyte and vascular interactions. So there's even a snake venom directed towards it. So my question is, why do the mice and the humans that have a deficiency in GP6 have such a mild bleeding phenotype? Do you have any mechanism behind it? Can you help me with that?
Highly esteemed opponent, thank you very much for your question. This is indeed a very good point, and it seems way too good to be true. This is true. It could be that during the process of hemostasis, the function of GP6, which is of course there, may be overtaken by other pathways. To give an example, I mean, we have the coagulation cascade, which is a very strong component in this occasion. So when we're bleeding, the coagulation cascade activates very fast, and not only produce fibrin, but also generate thrombin, which activate platelets. And this is just one of the examples. Then there are many receptors involved in the process, or for example, as soon as we have integrin activated, this could be already sufficient to stop bleeding. Mm. The process of thrombosis is instead slightly different. For instance, it could be in some occasions that we have an higher increase of the ligands that are necessary for GP6 to get fully activated. Therefore, in the process of thrombosis, it could simply be that we have a higher stimulation of this receptor respect to others, which is proven by the fact, for example, that when we have an atherosclerotic plaque, in some studies have shown that this is enriched with collagen type 1 and type 3, which are the collagens that are preferred by GP6, for example. But of course, GP6 is not always this magic tool, like with magic target that we can use. For example, there are evidence that it is also involved, it may be also involved in venous thrombosis, but it's probably, it is not being proposed yet as a target for it, to give an example. And yes, of course, there are many applications in which this could be interesting to be studied, for example.
Yeah, no. I think that the applications will have a lot of attention also in this round. I have another question on your chapter 4, where you, of course, present the human GP6 mice and the anti-GP6 antibodies. So I know the company Emfred. I think EMF1 is probably derived from Emfred. And it offers also a very well-known antibody, widely used antibody, R300. Are you familiar with R300?
Yes.
Well, I give it for the audience. That's an anti-GP1B alpha antibody. And you see it if you download the manual. You can even see it if you inject that antibody in mice, also at the same concentration or the same doses as you did. You get a prolonged depletion of platelets. So my question is, why, if you use GP6 antibodies, you only lose it from the surface? And if you use GP1B alpha antibodies, you completely deplete the platelets. Do you have any idea how that works?
So that's a very good point, and I do not have experimental evidence. However, we could speculate that, for example, GP6 is much less expressed on the platelet surface, and this could already be. So there is a published paper that shows the mechanism on how GP6 is depleted. And we could speculate that something similar may happen to other receptors. And based on this, I could speculate that the event of GP6 downregulation respect to that of GP1B downregulation may not be as hard for platelets respect to the how it is or when we treat them with GP1B. So GP1B antibodies. So this could be one of the explanations.
Okay, good. Then I have one final question on the half-lives of the antibodies, where you actually stress a lot. In the introduction, you stress it on page 29. You talk a lot about half-lives also in chapter 4. So why do you need a long half-life? I didn't understand it from the text. Why can't you have short half-life antibodies like the JAK1? And you may be even able to control the inhibition better if you have shorter half-lives.
This is absolutely true. And the thing is that there are already GP6 inhibitors in clinical trials. For example, if we take glensogimab, it's a Fab fragment, and it has a rather short half-life. It does not deplete the receptor, so it has a different function. So it could be that in models of diseases, this would work differently. However, we already have tools that have short half-life. We could also have the need of tools that have an increased half-life. This part of the half-life for inhibitors is something I really like to talk of, because if we have inhibitors with different half-life, we can use them for different things. So it doesn't mean that, perhaps I rephrase it. We could, I'm really interested into the high half-life of this antibody, because I think that it's really interesting that you can completely deplete the receptor from the surface and not have it there for quite some time, and you don't have to keep on treating the patients with something. And this I find interesting. Of course, it would be interesting to be able to modulate the depletion as well. an example is JAK1, but JAK1 does not have a shorter half-life, but more it has a shorter ability of, a smaller ability of removing. Yes.
Great that you mentioned that. So you want to say JAK1 depletes a little bit shorter than the EMF1, the one with the higher activity. Is that EMF? I think so.
Yes, yes.
Yeah. Okay, so you refer here to page 120, figure 1C versus figure page 131, 5C. But if I look at the times, I cannot really conclude, to be very honest, that the effects are so different between the two antibodies, because you've measured in figure 1C until day 5, and then the next measurement is at day 18, and the other one is at day 14, the latest, and then day 23. So, I think you should. Do you agree with me that you maybe need a little bit more time points in figure 1C to really be able to conclude that?
Yes, this is true, but in my opinion, the important thing into the treatment with JAK1 is not the half-life, but it's that it is not able to completely remove GP6 from the surface. Okay. So this is what I was interested here in this case, that we can inject an antibody and only partially remove it and remove the receptor, and that this partial removal is still sufficient to completely, to strongly reduce aggregate formation. Now, what is the evidence, the effect of this in thrombosis models or in a patient?
No, I would, also I see that the prorector completely agree with me, that I would like to give the word back and for the next opponent. Okay. Thank you very much.
Thank you very much, Professor Koene. Then the opposition will be continued by Professor Bender, who is present online. Welcome. He's professor of cardiovascular biology at the University of Würzburg. Welcome, Professor Bender.
Thank you very much. Dear candidate, also congratulations from my side to your thesis, which is very comprehensive, a lot of data, and yeah, a really nice work. So my first question is about, so the humanized mouse models are really a nice system because you are more to the human situation. However, when we look into the copy numbers, so for example, CLEC-2 has a 20 times higher copy number on mouse platelets compared to human platelets. And also the GP6 copy numbers varies between human and mouse platelets. So my question is, what about the signaling cascade? Because when we think about the SIC protein, it has 5 times more copy numbers in the mouse platelets compared to human platelets. So do you think maybe the SIC, for example, the SIC pool would shift to other receptors, and maybe you have in these mouse models a different signaling cascade for other receptors that you haven't looked at?
Is this working? Okay. Highly esteemed opponent, thank you very much for your question. So you raise a very good point. In some cases, there could be a chance that this may lead to differences between mouse and human. We trusted our humanized mouse model with the specific, and in this moment I'm talking of the humanized GP6, then we can also talk of the CLEC-2 mouse line, because we have shown that there does not seem to be a difference, at least between mouse and humanized mouse in every in vitro or ex vivo assay that we have performed regarding to platelet activation. Therefore, we took a model, which is the mouse model, and we implemented a human receptor in order to allow us to specifically study the effect of inhibitors of human GP6 in this case. However, to address whether this could be different, so the effect of different treatments or also of the function of GP6 could be different than in human human platelet, this needs to be addressed with different tests. But we also shown that these platelets tend to react in some ways when they're humanized similarly to human platelet. And I'm talking in the case, for example, of the reaction to convalescin. So these platelets can react to GP6 dependent signaling in a very similar manner respect to control mice or wild-type mice. However, when we treat these mice better, when we activate platelets using convalescin, we have instead an effect which is much more similar to the human platelets respect to that of wild-type platelets. And all these pieces of the puzzles allow us to understand that this mouse line seems to really be a good tool to test these antithrombotic drugs in vivo. Okay.
So I have a question to a figure on page 120 of your thesis. Do you have the book in front of you?
Yes.
Yes, okay. So it's regarding subfigure K and L. So I was wondering, a bit puzzled about this. So you inject the JAK1 antibody and you deplete, of course, the mouse GP6 completely from wild type mouse platelets, but also from the humanized heterozygous mice. When you look into the human GP6, you only get a partial reduction for the homozygous humanized mouse, but to a similar extent for the heterozygous mouse. So would you not expect to have a further reduction in the heterozygous humanized mouse models because the receptor density is already lower from the start?
Yeah, yes, and this is why we found this particularly interesting. So apparently, the effect of the antibody JAK1 or its effect is not dependent on receptor density, which is striking, but also quite particular. So independently of how much receptor you have on the platelet surface, it seems that this antibody, once injected, can only remove a certain percentage of the receptor. This antibody can only remove a certain percentage of the receptor for the platelet surface. For the precise reason, we need to do further investigation, and of course, this is why the paper is not yet published. But that's indeed one of the main points that we were quite puzzled by.
Okay, thank you. Then I have a completely different question. So to the chapter 5, the humanized CLEC-2 mice. So for the humanized CLEC-2 mice, you have a defect in the ferric chloride arterial model, which is of course different to the wild type mice. So can you speculate why you have this observation? Is this based on the ligand or of the copy number? Is it the signaling proteins that are different? What is your explanation for this observation?
So you could speculate a lot on this result, and that's definitely unexpected. There are some possible explanations. For example, so we have in this graph that if we have wild-type mice, so we have normal thrombus formation. If we have the humanized CLEC-2 mice, we have mostly non-clotting mice. And if we have the depletion of the humanized mice, we revert back to the wild-type mice, which is quite striking and is quite paradoxical. One explanation could be that human CLEC-2, but this is definitely just a speculation. The human CLEC-2 has somehow, in this case, inhibitory effect respect to the activatory effect of mouse CLEC-2, which is, I understand, hard to believe. However, if we remove human CLEC-2 from the surface of these mice, we have completely reversion of the phenotype. Therefore, there must be something that this receptor does on the platelet surface to give this effect. And so this is the main speculation that I can do at this moment. But definitely, this is something that further research need to be done.
Okay, thank you. So my next question is about the Maastricht chamber assays. So when you when you inhibit GP6 after the first 2 minutes, you don't see a bigger effect on plate adhesion, even on collagen only, and on thrombus formation, for example. So how do you explain this? Because GP6 was also discovered as a fibrin receptor. Would this contradict your results to this finding, or what is the explanation for this?
So, in my opinion, the point is that if we imagine the blood flow during the coagulation process as well, and we have GP6 that is one of the first actors. So we have GP1B, then we have GP6 that binds the collagen. What is collagen, which is the main ligand of this receptor, is immediately on the surface of the injured vessel, or in our case of the microfluidic system. Therefore, GP6 is strongly necessary to activate platelets in that moment. In a moment in which we have an active coagulation cascade, the end fibrin is already formed, so there is a thrombus that is already covered by fibrin. The inhibition via GP6 would be not sufficient to keep on reducing the further aggregate formation, because you have so many pathways in that moment that are ongoing. One example is the thrombin. Thrombin has been proved by the same article to be prolongedly active also at later phases. Therefore, the theory is that GP6 is very necessary at the beginning, as there is collagen, they need activation, but at later phases, its binding perhaps to fibrinogen or fibrin is not as necessary, as there are already, the platelets get activated by, for example, thrombin, and then the integrins get activated, and then at that point, the activation of platelet is already sufficient. However, this happens in case of fibrin deposition. If we have, for example, cases of where we don't have active coagulation, so the surface where there is no tissue factor, we will have a limited effect of GP6 inhibitors also in this case. And this proves that when there is the fibrin clot already formed, the GP6 inhibitors are not so necessary anymore. They cannot penetrate the aggregate, but also they're not strong enough to further block the aggregate formation. And this is also this. Sorry.
Okay. Thank you very much. How much time is left, dear Prorector?
No, I'm sorry. No more time. Thank you. Yeah, thank you very much, Professor Bender. The opposition will be continued by Professor Dandekar, Professor of Bioinformatics at the University of Würzburg, also present online.
Yeah. Thank you. I would also congratulate you on your nice presentation and this interesting thesis. And so I would now like to ask a question to the proposition 1. Maybe one of the Paromitsas can read this one.
Proposition number 1. The JAK1 binding epitope on human and mouse glycoprotein 6, GP6, has diverged during evolution, resulting in different functions. This thesis.
Yes, thank you. I found this a nice, intriguing result. And can you name then some of the different functions which evolved here?
So the most obvious, highly esteemed opponent, thank you for your question, I'm sorry. The main one that comes to my mind is that human GP6 has been proved to interact with fibrinogen and to allow spreading of platelets on a fibrinogen-coated surface, which is something that is not, at least so far, not experimentally proved for mouse GP6. This is also shown by the fact that if we take the humanized platelets, for example, we have increased spreading of them on the fibrinogen-coated surface respect to control mice. So this is the first and main modification that comes to my mind. But this proposition is like something important for the why do we use the humanized mouse models. This is not just the last thing. We have to consider also that these two receptors are not like they're on different chromosomes. They are a different degree of similarity both at the nucleotidic level and at the amino acid level. And this may lead to different functions, one of which now we proved is the antibody, is the effect due to the antibody JAK1. And we find interesting that this antibody has these two strong different functions, as it clearly has to bind the same epitope. But nonetheless, this causes a completely different effect. It's a paradoxical effect. And this can be explained only by the fact that even though the receptors are similar, they are not that similar. And this is why we really need to use the humanized, like not really need to use, but the humanized mouse models are definitely a good tool.
Then I would like to ask a question to proposition 2. And please read out proposition 2.
Proposition 2. In shear-dependent thrombus formation, human platelet thrombin receptors have a more persistent contribution than the collagen receptor GP6. This thesis.
Thank you. I think also this finding is rather interesting, but could you name some of the data on which this statement relies?
Yes, so the main one that I can tell is with one of the articles in chapter 3. I hope. Basically, we show that when we use inhibitors for for GP6 under flow in active coagulation, we have a limited effect of the receptor of the inhibition of GP6. So if we treat human blood with the antibody EMF1 in this case, we have a strong reduction of thrombus formation, aggregation, thrombostability, phosphatidylserine exposure, and therefore fibrin deposition. However, if we treat the human blood 2 minutes after the formation of thrombi has started, we completely lose the effect. However, if we do the same experiment, but with the inhibitors of the receptors PAR1 and PAR4, we have a prolongedly active effect, which means that the thrombin-dependent aggregate formation is required also at later phases. So we have still a reduction of thrombus formation. When we add 2 minutes after the PAR1 and 4 inhibitors, the thrombus formation suddenly reduces and grow very fast. But this does not happen at all with GP6 2 minutes after the formation of thrombi.
Okay, yeah, this is quite convincing and was definitely a lot of work. Now I have a final question to proposition 8.
Proposition number 8. Despite technological and medical advances of the last decades, cardiovascular diseases are still leading causes of death globally. Platelet research is fundamental for achieving further disease reduction and better treatment. Impact this thesis.
Yeah, thank you again. I have no doubt that the research impact of the thesis is quite strong, but can you allude a bit more how we could imagine now transfer to clinic?
Sorry, if I understood well, to transfer this to the clinic. The first thing is that after a long time of studying this receptor, I'm talking of GP6 in this case, we finally, scientific community, not me, finally came to the conclusion that this seems to really be a strong target for treating thrombotic events. For example, if we add, there are clinical trials showing already positive effect in ischemic stroke for the antibody glensogimab, or better, the Fab fragment glensogimab. And how could we use this research instead? Is first of all, we are showing a proposed treatment that is not yet been tested in clinical trials, and this is a treatment that differs from what has been already tried so far. as this shows that we can block the function of this receptor by completely eliminating it for several weeks without inducing major bleeding defects. Interestingly, it has already been shown also in different patients affected by immune thrombocytopenia that autoantibodies against human GP6 in patients remove the receptor from the platelet surface, showing that this that we have proved to happen in mouse may also be the case in human. And so to the question, how could we translate this in the clinic would be, first of all to do quite some study in animal models to test for the safety of this treatment, and secondly, therefore we could have a tool that could be used for chronic treatments more than for acute treatments, like in the case of glensojimab. And yeah, so I guess this is the main reason I can give in this moment.
Thank you very much, Professor Dandekar. We now move to Professor Elvers, Professor of Experimental Vascular Medicine at the Heinrich Heine University in Düsseldorf. Professor Elvers.
Thank you, Ms. Goldberg, candidate. So thank you very much for this brief overview of your results and the very comprehensive work. And I think this is really clinically very important and relevant. I have questions to chapter 3 and 4 mainly. Chapter 3, can you just comment on how important it is this to analyze the time-dependent contribution of different signaling pathways that contribute to thrombus formation, and what is the advantage of the multi-parameter approach of your flow chamber experiments?
Highly esteemed opponent, thank you very much for your question. So I believe that if I understood the question correctly, the relevance of this study in the treatment with these different receptors. So I find this study quite interesting, in my opinion, as it shows that the targeting of different receptors at different time points of thrombus formation have strongly different effects, which shows us why, for example, if we have already formed fibrin clot, it would be harder to use an inhibitor, for example, against GP6 in the clinic, unless we cope it together with something that has to first dismantle the fibrin clots. Like in this case, I always give the example of glensojimab, as this is the first target that give good clinical evidence so far. We have, in this case, the use of GP6 as an add-on therapy to the RTPA. And in this case, so our study proves why we would need to use this as an add-on therapy and not as a therapy itself in this case. Secondly, this study also shows, first of all, that we have a very useful system that can be used to specifically show the effect of the different receptor during the process of thrombus formation. To give the example is when we treated the human blood using tirofiban. We can see that when we use tirofiban on human blood at any given time point, we completely block the formation of thrombi. And how does this translate with what we know already? By the fact that inhibitors of the integrin alpha-2 beta-3 are very strong and may lead to very strong side effects as well. And this is why they are used only in some specific cases. And I believe that the system that we have developed can show very nicely the effect of this treatment. Something else that I can say about this study is also that the inhibitors of the PAR receptors, I believe this is also quite interesting, because when we inhibit both receptors at the same time, PAR1 and PAR4, we still have a very strong reduction of thrombus formation, but this reduction is not as strong as when we use, for example, integrin inhibitors. Or, for example, this also shows us that inhibitors of PAR1, PAR4, but also of the alpha-2 beta-3 do not have any effect on the coagulation. So, for example, if we treat the human blood with GP6 inhibitors, we have a complete abolishment of phosphatidylserine exposure, which means an abolishment of the procoagulant surface of platelets. And procoagulant platelets have been already associated with an increased risk of stroke, ischemic stroke in this case. And this shows us, again, why the treatment with GP6 may be beneficial in case of ischemic stroke. So these are some of the examples I can give in this moment.
Thank you very much. So I have 2 questions to chapter 4. The first one is if this has been analyzed, or if you have any idea about that. You just showed a GP6 low phenotype with JAK1 or JAK4. Do you know what the effects are in vivo with regard to thrombus formation?
Perhaps I forget before, so I apologize. Highly esteemed opponent, thank you for your question.
Maybe you can give a short answer.
Sorry.
Try to give a short answer.
Short answer, I'm sorry. So this is already under investigation. Therefore, I cannot give a clear direct answer on the effect in vivo, as it has been shown in the articles for the complete inhibition. We believe, though, that this would be interesting to assess, and to assess in also together with other treatments. So this is what we will do in the future.
That's fine. Have a question, or?
Only if it's a short question and a short answer, because otherwise we're running out of time.
Yeah, actually, perhaps you can just speculate about the advantage or disadvantages to block GP6 or to delete GP6 for antibody treatment in patients as a therapy.
So I believe I did not completely understand, though, the first part. I'm sorry. So the speculation of these antibodies in therapy, the ones in my thesis. Very short, we do not believe that we can use an antibody that is a GP6 enhancer in human, like JAK1. JAK1 is just an example of what can be done. In regard to the antibody EMF1, we know that this is, at least this thesis shown that so far, it's not provoking any increased issues in hemostasis, and that it inhibits very strongly thrombus formation. Therefore, we believe that such a therapy could be at least hypothesized.
Thank you, Professor Elvers. We now move to Dr. van der Vorst. He's researcher at the Department of Pathology at Maastricht University.
Yes, thank you very much. So dear candidate, first of all, also congratulations on this nice thesis, also for your supervising team. So I have many questions. I stick to a couple of them. I find it very interesting. And one of the interesting points was in chapter 3, where you then compare this time difference between treatment and 0 minutes or treatment at 2 minutes. Then you basically see that at 2 minutes you have a very massively different effect. What can you speculate about the reason? Is the effect just delayed, and will you observe it later, or did you miss your time window of treatment? And what's the reason then?
Esteemed opponent, thank you for your question. So what we did here was we selected the 2 minute time points in our system, as this showed us from a previous control experiment, that it generates limited amount of fibrin around the thrombi already, and that the thrombi are already therefore stable, however still small. And we wanted to see whether we would be able in premise to or to what extent inhibit further coagulation and platelet aggregation. Therefore, we selected this time point on base of this previous experiment. However, of course, it would be also interesting to test previous time points, for example, 1 minute. I believe that there are other studies that other studies that shown the effect of some inhibitors of GP6 at different time points in a system that is not exactly the same, but it's a similar one. At later time points would definitely also be interesting, but I believe that considering the results not really for studying the effect of GP6 inhibitors, but for example for PAR inhibitors and also for the effect of the tirofiban. In this case, we use tirofiban, so for alpha 2 beta 3 inhibitors. And I'm sorry, I don't know if you asked about the relevance of this in.
Yeah, and maybe especially. So you talked about, okay, in 2 minutes you have these small thrombi with fibrin. Do you think is this fibrin deposition then the reason why these antibodies are less effective? Are you then blocking the docking size of the antibody, or why are these antibodies less efficient?
So I definitely believe that this is the case, at least for the antibody EMF1, as we show that when we use the same treatment with the minus tissue factor coated surface, we have a disaggregation of the thrombi. And this is not just been proved by us, but also other articles before this shown that there is a disaggregation due to GP6 inhibitors. Therefore, yes, the inhibition of GP6, it's somewhat reduced in the moment you have a fibrin deposition. And this is probably because the thrombus is so stabilized and so retracted that it's very difficult for the antibody to enter within the thrombus. And therefore, if we would have an effect in this case, we could not see it when the fibrin is there.
Mm-hmm. And you now already mentioned it. Basically, if you then treat these thrombi at a bit later stage, you get this disaggregation of the thrombi again. And I think also in chapter 4 you also described this, that you have this transient reduction in blood flow, but then it comes back basically. And then you described this, okay, disaggregates, you get emboli formation. Do you foresee any difficulties with this then, if you think about this in a clinical setting? If you're then basically too late in treating the patients, will you not induce emboli formation?
So, embolus formation, I'm not sure, to be honest. What I can say is then that if we want to put this study in a more clinical setting, what it's telling us is that some inhibitors may be ineffective at later phases. So when we already have thrombus formed. So for example, if we could divide very simply like the treatment for thrombotic events in 2 ways. So either an acute or a chronic. And something that doesn't work when the fibrin is already there and when the thrombus is already there, it's not let's say, wouldn't be useful on its own in an acute situation. However, in a chronic situation, this could be really useful because in the moment we have, in this case, for example, collagen exposed, we don't allow the platelets to aggregate on it at a very fast rate. Or the thrombus is so unstable that this won't allow the generation of a thrombus, which then will cause the embolus to be formed. So embolus that is probably life-threatening, I would not imagine. Of course, this is not something I can exclude. However, again, I do always the same example, at least for the receptor GP6, so glensojumab. There are positive clinical trials used as an add-on therapy, and this showed that there is a benefit and not a reduction. And the benefit is probably due to the fact that once the RTPA helps removing fibrin from the thrombus, and GP6 probably switches off the further platelets that come there and the procoagulant surface of these platelets, then we have a positive, enhanced positive effect in this case. If we would use it on its own, I cannot say because no one has ever done it in the clinic, but the effect would most likely be reduced. But
so then the conclusion is basically we should treat as early as possible. Then you're always on the safe side. If you then think in this context, you say in the 0 minutes, there it is very effective, there it works. If we get this into the clinical setting, would you then not say, okay, in these high-risk patients groups, they don't have the thrombus event yet, but they have a high risk to get this, we already prophylactically start treating these patients? Would this be a good way, or do you see limitations in this approach?
So the prevention is always good. We already have drugs that prevent. So the reason why we do this is because we believe that the drugs that are currently used in this moment still are not completely perfect in this case. it would definitely be good to have a acute chronic treatment of these patients with these targets. The problem is, how do you chronically treat a patient with an antibody? It's like, at the moment, the way we have to treat patients with an antibody is to inject it, which is not something that people really love. So there are sides, let's say, in this case. So for example, these are still treatments that probably will be used in the near future. However, for the chronic treatments, there are still some challenges that needs to be overruled. And the reason why I was always stressing a lot over the lifespan of this treatment with the antibodies is that these strongly reduce the times you need to treat the patient with these drugs. Now, it does not have to be an antibody, even though it's so far only antibodies have been proved to remove GP6 from the surface. But this is something that it's definitely interesting. So you can remove a complete receptor, the platelets come back to the circulation, they're fine, but you have protection from thrombotic events. And
thank you very much. This is already a good answer. I will give the word back to the prorector. Thank you.
Thank you very much, Dr. van der Vorst. It's now time for the last opponent. It's Dr. Depperman. He's assistant professor at the Center of Thrombosis and Hemostasis at the University Medical Center in Mainz.
Thank you very much, Mrs. Prorector. Dear candidate, I would also like to congratulate you on your nice thesis work. And I have a question. In your introduction, you talk a lot about the different functions and important functions of GP6. So my question here would be, by suggesting to target GP6, would you not expect a lot of side effects?
Esteemed opponent, thank you very much for your question. The interesting point in targeting GP6 is that, first of all, it's expressed only on platelets and megakaryocytes, which already strongly reduces the side effects. Secondly, there are no evidence that show that there are like potent side effects when treating GP6. We do have, for example, there is the fear that targeting GP6 may increase, reduce vascular integrity, and this is also a main role of the receptor. However, there was a recent study, again, with the antibody glensojumab showing that it does not increase, for example, inflammatory bleeding when you target in human. Therefore, I would say that, of course, this is always something that needs to be assessed, but at the very moment, there is no clear evidence of very strong side effects due to the targeting of GP6 in clinical settings. And for example, it is also proved that patients that completely lack GP6 from the platelet surface do not have major bleeding complications. They do suffer of small bleeding diathesis, but they don't have major bleeding complications, and they live their life really happy. So,
okay, thank you very much. My next question is about. In your thesis, you write a lot about that it's a humanized mouse model, humanized GP6. My question is it really fully humanized? Or if not, what is lacking to make it a fully humanized GP6 mouse model, and how would you go about to do that?
So the mouse model for GP6 is genetically modified in order to add the cDNA of the receptor, the human receptor, in the genome of the mouse. Therefore, we have premature stop codon, and we have basically a disrupted gene in the mouse GP6, and therefore a full expression of human GP6 from the platelet surface. So from the side of the expression of the rate of expression of human and mouse GP6, we have a full humanization of the mouse. So the mouse expresses only human GP6. The things that could come to mind is that there are maybe some effectors that in the platelet that do not have the same exact function as between mouse platelet and human platelet. Therefore, if someone would speculate that the mouse is not completely humanized, this is the only point I would think about. For example, there are recent evidence showing that RAC1 may have a slightly different effect, a different function between mouse and human platelet. However, for the matter of the receptor, the mouse is fully humanized.
Okay, thank you. One question about the mode of action of the JAK antibodies. Could you just quickly recapitulate how do they deplete the GP6?
Yes, so there is already an article published, I believe in 2016, showing that the depletion of GP6 happens in the liver. And the reason why this happens, it has been shown to, or at least in the article I'm citing, it has been proved to happen via increased shedding of the receptor. Therefore, this is the current understanding of the mechanism. So what happens is that the platelets, once in contact with the antibody, localize in the liver, and there somehow there is a mechanism that increases the shedding of the receptor, probably due to the fact that the receptor is occupied. This could be one of the explanation, and therefore the cells only lose the receptor and come back to the circulation.
Okay. Do you also see the receptor on megakaryocytes?
The antibody.
Yes, sorry, the antibody.
I have not seen it myself. I believe that this has not been shown. However, there is like a belief that that's one reason why the lifespan of this treatment is so high, so long. Therefore, it could be that the antibody binds to the receptor.
Stefano Navarro, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and of your defense, and I request that you and your company await the results of our deliberations and our return in this room.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Stefano Navarro. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Heemskerk is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
We first get a promise. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Stefano Navarro, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and I fix it with the official seal of the university. Dear Dr. Navarro, dear Stefano, it's a real honor for me to be the first person to congratulate you with this double doctorate. And in the congratulations also I'd like to include your family, relatives and friends present here, or attending the ceremony online. At the University of Würzburg as well of Maastricht, you have been doing your best to compose and write these two universities theses and to obtain this double degree. Your thesis indeed is a masterpiece in 3 different ways, and all Corona members here present agreed that the defense was very good. Hence the bill handed over to you is well deserved and is the result of 4 years of hard scientific work. Dear Stefano, when looking back at 2018, when you applied for the ESR position in the TAPPERS program, I found your CV indicating highest levels for your bachelor and master degree at the universities of Naples, Federico too. So at that time, doing your bachelor and master, you have done already very well to get those degrees. Your thesis in Naples concerned whole exome sequencing and analysis of dried blood samples. And I can't remember it exactly, but I guess at the time we were some way triggered by a project on dried blood samples as an excellent preparation for a PhD project on functional blood platelets. Before handing over to your other supervisors who wish to praise you, a short notice in my view why your thesis is a threefold masterpiece. One is you managed to get a thesis approved by 2 universities in 2 different countries with quite different sets of regulations. For the audience to give an idea on the complexity of a double doctorate, it took several years before the University of Würzburg and Maastricht made it to a signed agreement for Stefano, and we have seen today that agreement worked actually quite well. Another masterpiece is the 3-way blood inlet chute system, made by you in a few weeks. In this respect, it's good to mention that before you arrived, it took several years to obtain a working 2-way inlet system from immediate mixing of blood samples and recalcification medium. And then you came to the lab and you increased the system into a 3-way inlet system, and you managed to use this masterpiece as the basis for a scientific paper in less than 3 months' works. So well done. And the main masterpiece of our course is laid in the work that you performed during most of the PhD time when working in the Steger and Niesland labs in Würzburg. You worked with transgenic mice, with new antibodies, and you performed many complex complementary in vivo, ex vivo, and in vitro experiments in a successful way. And not many PhD students have been able to do this. The result of this is an impressive thesis with front pages of the universities of both Würzburg and Maastricht. And now I hand over to your co-supervisor, Dr. Marijke.
Esteemed Dr. Navarro, dear Stefano, I'm also very happy and proud that I can be one of the first to congratulate you on obtaining your PhD. And I also want to include your girlfriend, Johanna, and your mother and your sister who are here, and also your other family and friends who are listening online. So you started the PhD project in September 2018 as ESR number 14 in the TAPAS consortium. And I went back to how it all started. And then I found your motivation letter. And from this I can quote, so that is what you wrote. The reason I'm going to apply for a PhD with TAPAS is that as a next step of my career, I'm looking for a new, fascinating experience in the research in order to improve my personal skills. So I hope that when you look back now on your whole PhD period, that it was indeed a fascinating experience for you. And what I can say for myself, that it was for sure a fascinating experience to work with you. And I also hope that you have been able to improve your personal skills as a researcher to your own satisfaction. But based on your excellent thesis and also on the outstanding defense today, I must conclude that you have for sure the personal skills to grow further into a great scientist. So how did Stefano perform as a PhD student in the lab? I'm sure that Professor Niesland will also say a few words on your time in Würzburg, because you spent the biggest part of your PhD over there. So I can only speak about your secondment in Maastricht. And how you performed in our lab can also be best illustrated by another quote from your motivation letter. You said, "My strongest personal advantages are high adaptability and communication skills." So, as you were already well trained in Würzburg, you indeed adapted yourself very well in our lab. You started to perform flow chamber experiments together with Frauke Swieringa to learn our protocols and find your way around the lab. And I think within 2 weeks you're already off on your own. And you managed to invent the three-way inlet system, as Professor Heemskerk already mentioned also, and you showed it in the presentation. And you worked basically non-stop in our lab during the 6 months you were in Maastricht. And in this time you managed to experimentally complete a whole research paper. So this illustrates that you are indeed a highly adaptable person who can communicate splendidly. You also fitted very well in our group and department because I think you are a very social person. And it did not take too long before you were introduced, I think, to the bouldering hall by the other PhD students, including your paranymph Claudia. So, and another nice example from your social skills presented itself during the ISTH conference in London last summer. It is tradition during the ISTH conference that the Dutch scientists meet up one evening in the bar to discuss science over some drinks, mostly containing alcohol. And it is from this meeting and tradition that I would like to share with you a story that was actually provided to me by your colleagues from Würzburg, Dr. Zoltan Nagy and Dr. David Stegner. So they wrote to me that apparently on the way home to the hotel after the Dutch party, after a few drinks, Stefano gave his talk in full under the underground, which was scheduled in the next meeting for the next morning. And then I can quote from Zoltan. His character further grew in his colleagues' eyes when he delivered his talk on the London Underground after the Dutch night on the ISTH 2022, a few hours before his scheduled talk the next morning. This showed that Stefano can not only do excellent research and be a highly social person, but can blend these two perfectly, as he delivered a very impressive talk the next morning. So I would like to end here by thanking all the members of the assessment committee, Professor Heijmans, Professor Bender, Professor Dandekar, Professor Elvers, Dr. Van der Vos and Dr. Depperman for their time to evaluate and approve the thesis. And also thank you for being here today and participate in the Corona. And a special word of thanks also to Professor Koene, who was very kind to step in today as a chair for the assessment committee. So dear Stefano, I wish you a lot of success in your scientific career and all the best for the future. Enjoy all the beautiful moments that are yet to come and start today, because today it's your PhD celebration.
Thank you.
Dear Dr. Navarro, also on behalf of Maastricht University, I would like to congratulate you with the degree you have acquired. Of course, I also like to congratulate your family, friends and colleagues present here and online, who undoubtedly supported you during your PhD journey, because a journey it has been. That's always the case. And I really hope that you look back on this period with a lot of satisfaction, also on today, the way you defended your thesis. I can tell you that the committee was very happy with the way you did it, so you can be addressed about that. I'd also like to congratulate your supervisor, Professor Nieswand, who is online, Professor Heemskerk, Dr. Herman and Dr. Kuipers, from Würzburg and from Maastricht. So you've been at a lot of different places. That must have taken a lot of adaptation, I already heard so. I'd like to thank the degree committee present here and present online. Thank you all on behalf of Maastricht University for your participation in this defense. And well, that makes us come to an end of this ceremony. I wish you a lot of success in your further career, and I hope you look back on this session with a lot of satisfaction. Yeah. Professor Nieswand, you wanted to say something?
Indeed. Can you hear me? Unfortunately, I've got a feedback on here. So since we are obviously very much over time, dear esteemed Dr. Navarro, is this the right word to approach you from now on? Dear Stefano, so I've not prepared a real speech like this, but I would just like to say that today I'm very proud of your development, what you have achieved, and at every time point it was really intriguing to see your excitement about science. And this already started when we had the interview day, and I looked it up. It was on May 23 in 2018. You were one of 3 candidates, and already on that day it became obvious that you wanted to have this position. You wanted to have this project, and you wanted to join exactly this consortium. And since then, you have developed into a real GP6 and also CLEC-2 expert. And I can tell all the people who listen now, you've included a lot of data into your thesis, but there is more to come. And I'm very happy that you will continue your work on platelets, at least for some time. And I really congratulate you and your family on achieving all the things that you presented today and on the things that will come. So it was at any time point a great pleasure to work with you and to have you in our laboratory, but of course also fueling the collaboration with Maastricht University and Johan's group. So with this, I would like to finish in order to get you to celebrate. And I look forward to having you back in Würzburg. Hopefully you manage to get there with not too much alcohol, so that you can go back to work as soon as you have celebrated. Thank you very much for being part of our group. And have a good time. Thank you very much.
Thank you very much, Professor Nieswand. Yeah, with that, we are going to close this ceremony.